So good morning, students. Yeah. Yes. Still be regular, okay. Don't be late. Fine. Anyway, so far we have discussed many things. A quick review of whatever we discussed so far. So what things we discussed? We discussed about DNA structure and G trans for theories. We discussed about Griffith's theory. Then we discussed about what else? Every theory. We also discussed about Michel and Chase experiment we discussed. Then we discussed about DNA structure. Then we also discussed about what else we discussed? That's it, right? And the role of contribution of who is she? Rosalind Franklin. So far we discussed this one. Then I want to discuss the most important thing, that is the DNA replication. So of course we also discussed about the central dogma of life, basics, central dogma of life, basics. Now listen. So what are the few things that we need to be considered when we are replicating a DNA? So why do we need to replicate the DNA? What is the purpose? Why DNA need to be replicated? Yes? What is the purpose? Yes? Why DNA need to be copied?
To keep the generation going.
Very good. To keep the generations going on, right? We have a single cell become two cells, and these cells are going on, right? The cells are dividing. How the cells are dividing? The cells are dividing because somehow the internet was connected. I guess Wi-Fi is there. Anyway, so the cells are dividing. That means the DNA was replicating. The DNA replicating indefinitely, leading to continuous generations. So now see, some few considerations include it should be rigid. It should be rigid. Zero errors. There should not be errors, or minimal errors must happen when copying. What else you guess? What other things must be there in a DNA replication? It should be strong. It should be zero errors or minimal errors while copying. What other things? It should be protected from environmental factors. It should be protected from environmental factors. Then what else? Any guesses? So then it should not be get affected by any stress. Okay, it should be continuous. It should be continuous, and it should have a long-term fertility. Fertility means survival. It should be able to survive for a long term. When I say long term, some billions of years. Okay. So these are the few things that we need to consider whenever the DNA was replicating. Then there are some candidates that we need to be considered. So there are some terms. Okay. So before discussing the terms, I just want to tell you that, see, we have a parent DNA. This is the parent DNA, right? Helical structure. 5' to 3', 3' to 5'. This is one DNA strand. Maybe it is helical, but I avoid it. Now, whenever it is undergoing a replication, what happens was, so it has a complementary strand. I don't know how to explain this. Imagine if my name was Vamsi, right? So the complementary strand, if this is one strand, the reverse strand will be small. So yeah, like this. This is how the DNA was arranged. One information from this way and other information from other way. Okay. So this is how the DNA information has been stored. So whenever I want to recreate my own another DNA, so I took VAMSI here as a one strand. Now I will start making a reverse strand. ILSMAB. Or similarly here we have ATGC reverse TAC connected. Okay. So, and we also know that out of 100% DNA, 97% of DNA is useless. Up to this portion is useless. Only some areas it can able to code a protein. Only in some portions of the DNA it can able to code a protein. Maybe that can be a HB or anything. So these regions are called as genes. These regions are called as genes. So some of the basics include. So if I need to create a hemoglobin protein, that particular part of DNA will be unwinded. First, that particular part of the DNA will be unwinded. So first we need to unwind the DNA. We need to unwind the DNA. Right? After unwinding the DNA, what we are doing? Now, after unwinding, we will initiate the replication. So we have unwinded. After unwinding, we are going to make a template. So this is the unwinded portion. This is called as replication 4. The place where the DNA was unwinded, where it's going to take some replication, some baby strands. That region is called as replication 4. Replication 4. So now we have our replication 4. Now here we have two parental DNA strands. Now a primer will be attached to initiate the DNA replication. So now writing one more thing. So here I wrote replication 4. You know what is replication 4. Then a primer was attached. It's a small strand of RNA that will initiate the elongation of DNA. So this is called as primer. So now you know what is the primer. So unwinding will be done by helicase. So there are some enzymes. Helicase is an enzyme that will unwind the DNA. DNA will be unwinded by the helicase. Then the region of replication is called replication 4. Then we have a primer. Primer will initiate the process. Okay. The next one will be after primer. The primer will be elongated. The primer from the primer region, the other strand will be created. Okay. That is called as replication. Okay, polymerization. So now we have an enzyme called polymerase. All enzymes will end with AS. Okay. This polymerase will elongate the DNA, elongate the strand to be short. Okay, elongate the strand. After polymerizing, what happens? See, after polymerizing, the strand came. This strand came and this strand came. So both the strands will make a baby DNA. Then next one. See, this portion should be rewinded. Wherever replication 4 happens, that portion should be rewinded. If not, that becomes a mutation. That gene should be again preserved there. So it should be rewinded. It should be rewinded. Okay. Now we got to baby the DNA strands, and the process will go on. Now it looks very simple, but there are few things that we need to be considered. Whenever the DNA was unwinded, there is a chance, just like a spring, it can come back to its original location. After we are extending the spring, it can back to original position. So there are some proteins that will hold this position. So there are many things that are embedded in this process. Now I will give you a simple classification. First we will write all the things involved in replication, then we will go further to discuss individual things. Okay. So right now, broadly categorizing, DNA replication has 5 steps. So steps of DNA replication. Steps of DNA replication. Write on this table. This table is very important. Steps of DNA replication. These steps are broadly categorized into 5 major steps. What are those 5? We will see. The first one, initiation. Replication initiation. Okay. The first phase is initiation. Initiation. Second, primer synthesis. Primer synthesis. Third step, elongation. elongation. Then, fourth step, removal of primer removal and gap filling. Primer removal and gap filling. Of course, after replication completion, we need to remove this primer and we need to refill the gap and we need to rewide it. So primer removal and gap filling is the fourth step. Primer removal and the gap should be refilled with the appropriate material. Then last and final, termination of the replication. The termination of the replication. Termination of replication. Termination of replication. Right? So, so these are the 5 major steps. Initiation of the replication, primer synthesis, then primer elongation, then removal of the primers and termination of the replication. Right? These are the 5 major steps. Now, right now, in initiation, we have 2 steps. In initiation, we have 2 sub-steps. Can I erase this portion?
Yes, sir.
Yeah. In initiation, we have 2 steps. 1 second. Yeah. Right now, in initiation, we have 2 steps. They are origin of replication. Origin of replication. That means in what part of the DNA the replication should happen. By the way, there are some differences between prokaryotes and eukaryotes. In prokaryotes, that means in bacteria, only one replication form will be opened. But in eukaryotes, multiple replication forms will be opened in the same DNA. So here one replication happens, there one happens, somewhere else happens in the same strand of DNA. That is possible only in the eukaryotes. But there is one disadvantage in eukaryotes. The disadvantage is the replication cycle was very complicated and it is very slow compared to the. You are very late, students. Anyway, sit in the back. See, compared to eukaryotes, prokaryote replication is fast because their DNA is simple DNA. But eukaryotes are complicated DNA, so it takes obviously lengthy time. So in initiation, we have 2 things: origin of replication. And second one, unwinding of DNA. Unwinding of DNA. Obviously, the DNA must be unwinded, right? So, in the initiation, we have origin and unwinding of the DNA. After unwinding of DNA, after DNA unwinding, 3 enzymes will play a role. There are 3 key events happening. The key events are helicase activity. Helicase activity. What does the helicase do? Helicase will unwind the DNA. Helicase will unwind the DNA. Then second one, SSB proteins involvement. SSB, write down the full form of SSB. Single strand binding proteins. Single strand binding proteins. The single strand binding proteins will be coming into action. I will tell one of the single strand protein bindings. Single strand binding proteins, what they will do is, see, after helicase unwinding the DNA, see, the helicase unwinded the DNA. Now, these SSB proteins will be, they will stick here strongly. They won't allow the DNA to re-align, to rewind. That rewinding has a technical word that is called re-annealing. SSBs will prevent re-annealing. Re-annealing means within the bracket rewinding of DNA. Rewinding of DNA. That is the action of SSB. The single stranded binding proteins will prevent rewinding of the DNA in a nutshell. Clear?
So what is the helicase? Helicase?
Helicase will unwind the DNA. It will unwind the DNA. See, helicase opened the DNA. Now these SSB proteins are keeping the unwinded DNA in its place. They will prevent rewinding. Okay. Maybe helicase SSBs can be stated like a car jumper. That we will lift the car there. So now that jack will prevent collapsing of the car. Similarly, SSDs will prevent, act like a car jacks. They will keep here in its place. Okay, yeah. Next one, topoisomerase. Oh, I don't know so far. This is topoisomerase. Just write down topoisomerase. So what this enzyme doing means it relieves supercoiling by cutting and rejoining DNA strands. It relieves supercoiling. They say that it will relieve supercoiling by cutting and rejoining DNA. By cutting and rejoining of DNA. Supercoiling. How can I tell you supercoiling is? You just imagine I have a long strands of springs, okay, and during this replication or simply I throw the springs on the floor. Now the springs will unwind and they will entangle together, right? That entanglement is called as supercoiling. If that entanglement happened, the only way what we can do is we will remove this portion and we will discard it. We will remove the entangled part of the DNA and we will proceed with a good portion of the DNA. So this topoisomerase will take care of that activity. Topoisomerase will relieve supercoiling by cutting and rejoining of DNA. Wherever the supercoiling happens, the excessive entanglement happens, that portion will be deleted and other portion will be rejoined. Okay? So that is the activity of topoisomerase. Topoisomerase will reduce the supercoiling of DNA by cutting and rejoining of DNA. So these are the major events happen in the initiation of the DNA or replication. Clear? Next one, primer synthesis. It's a straightforward thing. Now there is a short primer, complementary strand to the initiation. Primer will initiate the replication, by the way. So a small complementary strand will be attached and it will start elongating the required portion of the DNA. So primer synthesis. Primer will initiate the synthesis. There is nothing much to say. So it's a small piece of RNA, a small strand of RNA. By the way, I am not going back. There are some values here. It will be around 20 to 40 base pairs, the length of each primer. That depends. And each primer has its own temperatures. So primer will initiate the synthesis of the complementary strand. Complementary strand is the opposite strand of the parent DNA. Okay. Yeah. So primer synthesis. After primer synthesis, elongation of the DNA. Primer will initiate the synthesis of complementary strands. Primer will initiate the synthesis of complementary strand. Got it? Yeah. So now the primer started synthesizing the complementary strand. But that is not easy. Complementary strand should be synthesized by a polymerase. There is an enzyme that will polymerase, that will make the opposite structures connecting it to the A means P, G means C. Now coming to the elongation. In elongation we have four key events. Right now in elongation we have four key events. One DNA polymerase activity. 2. I hope you know what does the polymerase do. Polymerase will add a complementary base pair to the parent strand. It will add a complementary base pair. For example, here we have ATGC. What here it will add? This is the polymerase. It will add A means T. Again T. A. G means C. C means G. So it will have the opposite complementary things. Okay. Now keep in mind, polymerase always works in the direction of 5' to 3'. Keep in mind, polymerase always works from the direction of 5' to 3'. This is the movement. Its movement is always towards 3', from 5' and 5' and to 3'. That means replication happens like this. This is 5' to 3' this way, and another replication happens from this way. One strand prepared in that way, and other strand will be prepared this way. Because here this is 5' to 3', right? So this is how the replication happens. So it's very complicated. You are seeing one was going this way, one was going this way. Now keep in mind, there is a rule I forgot. Maybe I will get in the future notes. See, the 5' to 3' is the fastest method of doing. But from 3' to 5', there are two strands. During replication, there is a leading strand. That means the strand that is making very fastly, and there is one more strand, one lagging strand. Regarding this leading and lagging, we'll see later. One strand will be synthesized slowly, and another strand will be synthesized very fastly. Now, there is another problem. When we are making a slow strand, this slow strand will also make some fragments, a piece of unwanted DNA fragments. They are called as Okazaki fragments. In slow strand, some fragments of DNA will be lost, and those fragments are called as Okazaki fragments. Regarding these Okazaki fragments, we will discuss in future. Okay, just keep in mind this word. One leading strand, one lagging strand. Lagging strand makes some Okazaki fragments, and we will see later. Clear so far? So that is the activity of the DNA polymerase. So now we know what is DNA polymerase. It is polymerizing, it is adding complementary strands. Second one, as I said, see leading strand and lagging strand. So yeah, leading strand formation, and the third one, lagging strand formation. Lagging strand will lead to a fragments called Okazaki fragments. Okazaki fragments. Okay. See, if you can't understand my writing, don't worry. It's just for abstract, and I will give you detailed notes later. Okay, just for understanding. So now we have the DNA polymerase. It makes the two strands. One is leading strand and one is lagging strand. Lagging strand will make small fragments called Okazaki fragments. Okay. Then last and final step for this one. This is called as sliding clamp. What is this sliding clamp? Holds DNA polymerase in place for efficient. Okay. Sliding clamp. So there is one more thing involving here, that is called sliding clamp. Right now, sliding clamp. Sliding clamp. So what does this sliding clamp do? You know ropeway, right? Ropeway. We have a rope and there will be a basket hanging to this one. Right, ropeway, you know, right? There will be rope and you can sit here. What trees? Right? Yeah, house. Then what I was trying to say is this basket connected to the rope by a clamp, right? So what is the purpose of this clamp? This clamp will smoothen the journey. It will enhance the smooth movement of the basket on the rope. Similarly, when our polymerase enzyme connected to the DNA strand, it was connected by a clamp called sliding clamp. Sliding clamp will facilitate the smooth movement of the polymerase enzyme over the DNA strand. Sliding clamp will smoothen the movement of DNA polymerase for an efficient elongation. I hope you know what is elongation here. The complementary strand elongating, right? So that is called the purpose of sliding clamp. Over. So far we have understood what is initiation, what is primer synthesis. Then we discussed about what is elongation. In elongation, we have DNA polymerase activity, leading strand, lagging strand, Okazaki fragments, and the clamp activity. Then coming to the primer removal. Yeah. See. Now, two complementary strands are prepared. See, this strand was prepared, this strand was prepared. After that, what we will do? We will remove the primer. And we will fill the gap. Okay, we will remove the primer and we will fill the gap. So here, three activities are going on. You can read water. Yeah. In primary model gap filling, write down 3 series of key events happening here. 3 series of key events happening in primary model gap filling. The events are RNAS2. So what is this RNAS2? RNAS2. It's an enzyme RNAS2. So this will remove the primers. RNAS2 will remove primers. It will remove primers. RNAS2 will remove the primers. RNAS2 remove the primers. Okay. Next one. We have one more thing. DNA polymerase 1. DNA polymerase 1. So what is the purpose of this? It will fill the gaps with DNA nucleotides. It fills gaps with DNA nucleotides. It fills gaps with DNA nucleotides. So you know the difference between tide and side. So entire tide will be replaced by DNA polymerase. It will fill the gaps with DNA nucleotides. Then last and final, DNA ligase, very important. Very important. Helicase will unbind, whereas ligase will glue it. Ligase will bind it. Always, whenever you get the word ligase always binding something. Okay? DNA ligase. It will rebind. DNA ligase will join the Okazaki fragments by forming phosphate bonds. Just write down, I will discuss Okazaki fragments later as well, right? So DNA ligase joins the Okazaki fragments by forming phosphate bonds. DNA ligase joins the Okazaki fragments by joining the phosphate bonds. Fine. These are the major steps in removal and gap filling. Okay. And last and final, termination. In termination, 2 key events are happening. Shall I write here termination? In termination, 2 key events are happening. The key events are telomerase replication in eukaryotes. Telomerase replication. 1. Telomerase replication. Okay. And 4th, convergence. See students, 4th, convergence. One beautiful thing is there in this one. Actually, I need to discuss many things, but I don't have time. See, this is the chromosome. This is the centromere. This is pre-arm and this is Q-arm. Or simply this is the short arm and this is the long arm. Okay. See, whenever replication happening, or obviously there is some usage of DNA, right? Some piece of DNA might be lost because of Okazaki fragments. Something can be missed. Now, at the end of the chromosomes, we have something called telomerase. Telomerase is the portion. Now they are saying that what we observed was, in the children, telomerase will be around 10 centimeters. In the teenagers, telomerase will be around 5 centimeters. When we are getting old, telomerase will become 1 centimeter to 0.05. That indicates telomerase is the age factor. The more we are aging, the more we are losing the telomerase. Actually, telomerase acting like preserving. It's acting like a caps on the both the ends to maintain the DNA. So, but eventually we are losing the caps of the chromosome. So that is why we have a dedicated enzyme to rebuild the telomerase. But it is not so efficient, I guess. So we have this is only present in eukaryotes. In termination, after completion of the DNA, after completion of the daughter strands, now again it is trying to resynthesize the telomerase to preserve the DNA in the chromosome. So this is only present in the eukaryotes but not in prokaryotes. The reason is in prokaryotes they have circular DNA, open naked DNA, but in eukaryotes we have a protected nucleosome and inside the nucleus we have. And fourth, convergence. Replication ends with when four meet. Okay. Fourth, convergence means replication ends when both the four meets. Both the four meets. What do you mean by four here? In the beginning I said that this is for replication 4. So again they both will be meet and the replication will be completed. That's it. So these are the 5 key events in the DNA replication. The events are initiation, primer synthesis, primer elongation, primer removal and filling the gaps, and termination of the replication. And there are involvement of multiple enzymes that include helicase. What is the purpose of helicase? To unbind the DNA. What is the purpose of single-stranded binding proteins? Unbind the portion in place. So then what is the role of polymerase? It will elongate. What are the types of strands we prepare? Leading strand and lagging strand. What are the outcome of lagging strands? Okazaki fragments. What enzyme will reconnect the Okazaki fragments back to the parent strand? I said no. DNA ligase. Then what is the role of clamp, sliding clamp? What is the role of sliding clamp? For the smooth movement of the polymerase over the parent strand. What are the 5 key steps in DNA replication? Initiation, primer synthesis, primer elongation, primer removal and gap filling, and termination of the replication. What is the role of telomeres? What is the role of telomerase? To regenerate the telomere length. What is the significance of telomere? Telomere will preserve the DNA in its place. Through age, we will lose the telomeres. So there is some research going on. If we want to live longer, then we need to protect our telomeres. Is there any drugs or is there any methods of preserving the telomeres? So that research is going on. So there is one billionaire who wants to live for next 100 to 200 years, and he is working on all these things. He's spending a lot. Anyway, so this is all about the DNA replication. Let us see what the textbooks or the theory says. Let us deep dive somewhat. Okay. Yes, let us see the material. Some boring things. Usually I don't like PPTs. Anyway, I need to tell you one thing, students. Our human DNA replication is called as semi-conservative DNA replication. What is the meaning of semi-conservative DNA replication? Right now, see, our humans have a semi-conservative replication. Semi-conservative means, see, one parent strand will make, this is the, so this is the original strand and this is the daughter strand. This is the original strand and this is the daughter strand. So one parent DNA will be there and one daughter DNA will be there. Such kind of replication is called as semi-conservative replication. I will show you. See here. So this is the semi-conservative. One parent, one daughter. One parent, one daughter. Strands. Okay. Now, if any two daughter strands replicated, then they will make an entire new set of their own genes. This is called as semi-conservative. Whereas in a conservative model, the parent strands will be intact. They won't be disturbed. They will make opposite pairs, 2 copies of baby strands. Okay, this is called conservative. And we have another one. This is called as dispersive. In the same strand, some portion will be new one, some parent. Some parent, some new. Some parent, some new. So both things will be there in the all the strands. This is called as dispersive. Usually dispersive is seen among plants. So that is why in plants we can make multiple varieties actually. Do you know that in plant we can combine a mango with any other fruit. A mango with a similar genera of fruit. What we can take? Guava. So you can make both the guava tasted mango also. Because in plants they will accept the genes. That's not possible in mammals. But among plants they can accept the various other genes also. Because they are doing these things. So you will get a guava-ish, mango flavor. A combined hybrid fruit. So that is possible in the plants but not in the mammals or humans. Okay. So semi-conservative model is our model of replication. Yeah. So that's it. Okay. So in semi-conservative we have parent DNA and we have two daughter DNA models. Okay, during synthesis. So in a nutshell, this is what's happening. Very basic animation showing say replication 4 and it is keep on elongating. One leading strand, one lagging strand, and 3' to 5', 5' to 3'. So this is the simplest story. Now I just want to tell you one beauty, students. All these things happening. I just want to give one example of a red blood cell synthesizing hemoglobin. Can you guess, students? How many molecules of hemoglobin can be prepared by a red blood cell per second? Per second, how many hemoglobin molecules can be prepared? Can you guess? By a single red blood cell. 1 hemoglobin, 2 hemoglobin molecules. Any guesses? Believe it or not, it is 20,000 hemoglobin molecules every second. 20,000 molecules per second. That means see the fast, how quick they are. How fast this replication happening in the body. Because of this only we are sustaining, we are living. Millions of cells are producing every second to keep us alive. Millions of cells are dying. Not millions, trillions. Okay. Yeah. So this is a short animation to discuss the whole story of DNA replication. So we have a
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands.